My background is in mouse genetics, molecular biology, and genome engineering. So since 2017, I've applied this expertise to modeling and studying neurodegeneration. And in 2017, I joined an institute called MRC Harwell, working with Professor Lizzie Fisher at the Institute of Neurology at University College London, UCL. And our primary goal during that time was to engineer refined models for MND research. Specifically, we used genome engineering to replace mouse genes with human genes, and specifically human genes linked to MND, as well as inserting pathogenic mutations linked to different genetic forms of MND. And then in 2022, I moved to UCL, and I've been running a program of research at the MRC Prion unit at UCL. So this project is to advance therapeutics for a genetic form of MND caused by mutations in a gene called FUS. And it's using the FUS humanized mice that myself and colleagues engineered at Harwell. And we're testing a class of therapeutic called antisense oligonucleotides, ASOs, which are essentially short synthetic compounds quite similar to DNA and RNA. And what they do is they bind to a target gene product and block or sometimes modify production of the protein linked to that gene. So in the case of FUS, in which mutations cause the FUS protein to go to the wrong place in the cell, and essentially that protein becomes sticky and aggregates and ultimately causes motor neuron loss. The aim here is to reduce levels of that gene, reduce levels of that protein to try to limit the damage. And it's important for this project to highlight that it's bringing together different expertise in different areas. So from my side, we have the engineered mouse models and the expertise in live animal work, and our important collaborative partners at Murdoch University and the Perron Institute in Perth, Australia, who have developed and have expertise in these new ASO treatments, as well as expertise in human stem cell models, which will be important for understanding how human cells respond to these drugs. Gene therapy is what we call a precision medicine. And it's a game changer, I think, because instead of targeting the symptoms of a disease, which is how most small molecule drugs work, gene therapy takes advantage of our strong understanding of the genetics in different patients to target the root cause. So gene therapies for MND are essentially targeting these genes depending on the genetics of the individual patient, and genes that produce these rogue proteins in the brain or spinal cord. And as I said earlier, it's trying to lower these proteins to prevent their toxicity and prevent neuronal loss. Ultimately, it's important because there are very few approved treatments for MND. And part of that is due to the diverse genetics of MND. So we need to develop treatments against a range of targets. So there's still a way to go to develop these drugs, as well as I said earlier, as well as continued refinement to make them more effective. We're relatively early in the process. My colleagues in Australia have designed these new ASO drugs, and so we're aiming for proof of concept in a living organism in mice. And so the primary question we have is, can we lower levels of FUS safely and effectively in these models before we then take the next step? Yeah, the impact will hopefully offer new treatment options for patients with FUS mutations, but also think the findings from this work will offer insight towards developing similar treatments for other genetic forms of MND and beyond that for the wider neurodegeneration field.